kills it. Because the one thing that can destroy tuberculosis in a matter of seconds is UV. So one thing that's its weakness. Okay? So here you can see there's a gowning room, they're calling it an ante room, entry or whatever. You're gonna come in here, you're gonna gown up and you're gonna go in here and you're gonna protect yourself from the patient. Because the patient has an infectious disease. You'll be wearing a mask. You've seen the masks that kind of look like little duck bills. Those are anti-tuberculosis masks, right? And you should wear those if you're going to be working with a TB patient, and some of you will be working with TB patients, right? Okay? Anybody have any questions? Okay. I'm done with control. We're going on to talk about DNA and RNA biology. Okay. This morning, when I was getting ready, I put my pants on, and I felt something crawling on my leg. I'm like, uh-oh, what is that? So I pulled up my pant leg, and a scorpion landed on my foot.
Oh my God.
Here it is. That's a bark scorpion.
Is it alive?
It was alive, but I think it's dead now. One of the students smushed it, so it's kind of smushed. But if y'all want to look at it, how many people? It's in a bag, it can't get to you. But the stinger is still there, and it's still venomous. Do not touch the stinger. But if you want to look at it, here it is. Anybody want to look at it? Don't open the bag. It's a bark scorpion. It's small, but when it was crawling on my foot, it looked a lot bigger than it was. Like, oh my God! I usually try to take them up and put them outside, but I said I'd bring it in so y'all could see it. I think Justine wanted to see it also. It's a bark scorpion. I've been stung like several times. If they sting you, it's like a bee sting.
So it's not poisonous?
It's venomous, but it won't kill you. It just feels like a bee sting.
No.
You want to take a picture and show your boyfriend?
I know.
Okay. I think they're pretty cool. I like scorpions. I usually take them and put them outside because they're good critters. They kill roaches. Anything that kills a roach is good. And I thought I'd share my story with you before we got started. I don't think it's alive anymore. It hasn't moved in a couple of. Oh, maybe it is alive. Huh. We can do it later. Give a little resuscitation. I'll let it go later. Okay. We're not going to talk genetics. We're going to talk about how DNA and RNA works. That's it. If you had 1308 properly, this is just a review. Okay? I'm not going to get crazy on you. Okay, so let's talk about this. Right? So genetics as a whole is interested in how information in nucleic acids are expressed. And so the nucleic acids we're going to talk about are DNA and RNA. That's it. How the nucleic acids are duplicated and replicated and then transmitted to progeny. Anything that comes from our cells is progeny. So our skin cells, when they become two cells, the two skin cells are progeny. If we have children, like two daughters, then the two daughters are our progeny. Okay? And then how these processes account for the characteristics of the progeny, right? Why we look like we do, right? Because we're a combination of both of our parents' genetic code. Okay? There are two types of nucleic acids, right? And all nucleic acids are the material of heredity. They're large molecules and they're acidic by nature, and they're associated with the nuclear area of the cell. So if it's a eukaryotic, they're in the nucleus. If it's a prokaryotic, they're in what we call the nucleus. It's the free-floating DNA in the cytoplasm. Remember, bacteria are easy to study because they only have one chromosome, where we, as humans, have 23 pairs or 46 total. Anything that undergoes mitosis has to have an even number of chromosomes. Bacteria don't, because they undergo binary fission. Okay? We're going to use the bacteria as a model because they're easy to understand and they're easy to talk about. There are two types, there are several different types of nucleic acids. The only two we're gonna talk about is DNA and RNA, okay? So we think about that for a moment, right? So DNA, deoxyribonucleic acids, is responsible for all cellular activity. Anything that goes on a cell, it's a function of DNA. It's what makes it go, right? It directs the production of proteins, so under its direction, proteins are made by RNA. It's double-stranded and helical. It's maintained by hydrogen bonds. So 1 or 2 or 3 hydrogen bonds weak, but thousands or millions of them very strong. So DNA is very, very stable. It can survive temperatures as high as 70 degrees Celsius for extended periods of time. Where our body's at 37, so it's almost twice that, right? It can withstand high salt concentrations in acidic environments. There are cases right now being looked at again that the crimes happened in the 80s, where the DNA is still around in specimens or in evidence, and they're taking that, and what they're finding out in some cases, they put the wrong person in jail. So the wrong person has been in jail, prison for 40 plus years, right? Some of you, although all of you are not probably old enough to know this, but the yogurt shop murders that happened here in Austin, they just got solved because there was a detective that was doing a review. The person that died, they were looking at the review of what he had done, and what they found out, and this is one of the Carolinas, I don't remember which one of them, and they looked at the actual patterns that he used to kill some people, and he found them very similar to what happened in Austin, in the yogurt shop. And so he contacted the detective here. They were retired, but somebody else kind of held on to the cold case, and they sent them the information. They took the DNA in an independent laboratory and compared to the DNA that was on file for this guy that had died, and it was an exact match. This guy had committed both crimes, but he got away with this one. They caught him over there. Well, he's gone now, but it ruined it not only ruined the 4 young ladies' lives, but it ruined 3 other people's lives who were very young and made real simple mistakes when they were being questioned, right? And they implicated themselves as potential, and then they got thrown in jail. One of them got thrown in jail for a little while, but then got exonerated later. It's amazing. O.J. Simpson, how many people know about that guy? He did it.
Yeah.
The DNA evidence says he did it.
But the glow in the food.
How do you know that? You're so young.
I watched it. It rained
Well,
and it
the problem with it is that the jury of his peers did not understand what DNA was, and the prosecutor didn't do a good job of explaining it. But Johnny Cochran did a really good job of throwing shade on anything they presented when he said, if the glove don't fit, you must have quit. And that's all that the jury remembered. And it wasn't like days and days of liberation. It was like an hour, and then they came back and said, he's not guilty. I'm like, oh my God. Yeah. Nowadays, we have experts like my wife who goes in and explains what DNA is and how it works. She doesn't do it anymore. She hated that part of her job. That's what made her retire. And she hated going to court because the lawyers were never interested in the truth. They were only interested in trying to make you make a mistake so they could get their person off. Yep.
I watched your lecture and I remember you saying that too. But I was just wondering, did she like see a lot of people that she knew felt were guilty or felt strongly that were guilty
With my wife?
that got away with it, or did she not sit through the whole thing? She was just kind of.
Well, no, she sat through the whole thing because there was always a chance she was going to be called back. So she was not allowed to leave until the trial was over. But she worked on the animal side of law enforcement. She didn't work with. She said, "I will not work with humans. I don't want to have to go and pick samples off of somebody who's passed away."
Like illegal hunting or something?
Yeah, if somebody says I killed a hog, and then let's see, they take a little bit of all they need is a little bit of blood or a little bit of hair, and my wife could tell you what it was, how old it was, and was it a male or female.
That's cool. So she's in forensics.
She was a forensic scientist.
Wow.
Way smarter than I am. I can't hold a
I
straw.
wonder how those conversations are at home.
Yeah.
They get really terse sometimes, but a lot of times they're just kind of like people look at each other and go, they know what the hell they're talking about. But she's pretty cool. I like her. Okay. Anybody have questions about DNA? We're gonna be talking about it a lot in just a minute. The other one is RNA. And there's like 40 different types of RNA. We're only gonna talk about 3. And the 3 different types of RNA we're gonna talk about is messenger RNA, transfer RNA, and ribosomal RNA. You've already heard the term ribosomal RNA because it's used in a lot of different methodologies. It's used forensically, it's used in phylogenetic studies, it's used in a bunch of different things, right? But they all are important. Messenger RNA, its main function is to go and make a copy of DNA in RNA language. Does anybody know when I say copy DNA in RNA language, does anybody know what that means?
Transcription or.
Yeah, so when you transcribe it, you take a copy of it in a complementary form, but there is no thymine in RNA, so it gets replaced with uracil. I'll explain that in a minute, but that's what that means. Once that happens, then the messenger RNA goes to wherever the ribosome is, and it complexes with the ribosome, right? So it makes this kind of little complex. I mean, let's see. Who's doing, let's see, metronidazole, who's doing levaquin, and who's doing Bactrim? All of them are related to stopping DNA somehow or another, right? And somebody else doing tetracycline or erythromycin, that also works with the ribosome. So the ribosome is set up so you have a large subunit, you have a small subunit. It always kind of looks like a catcher's mitt to me. And then you have the messenger RNA come in and it complexes with it from a 5' to 3' direction, right? And when that happens, then the proteins are made. And we're going to talk about how that happens from a real high level. We're not going to get all nitty gritty in there. We're just going to do it at a very high level. Okay? So messenger RNA makes a copy, it complexes with the ribosome, and then transfer RNA, kind of to me always looks like a little firecracker. So these are what transfer RNA look like. And they're carrying an amino acid, whatever that amino acid is. But what they do is they come in and they place the amino acid in a chain, right? So you have a growing polypeptide chain that eventually is going to be folded by the ribosomal RNA, and you're going to have a protein. And what establishes the functionality of protein? The shape. So this is how this happens, right? Now, in a very simplistic way, I'm explaining it at a very high level. Now, I'm not getting nitty gritty. There's a lot more going on here, but we're going to be talking about some of this stuff.
Do we need to know like the codons and like anticodons?
Yes, you will, and I'm gonna get there and show you in just a minute. And since you already stole my magic, I'm gonna give you my favorite codon table now, but you won't have to use this till later, and I will give you one of these on the exam. Okay, you won't have to memorize it. Okay, there's no reason to. It's just mean if I did that, man.
Some of them, yeah.
You can tell the story to your daughters tonight.
I still did this in high school. You have to memorize that.
Okay, so transfer RNA is responsible for translation. It takes a look at the information that's a messenger RNA, and then it puts in place an amino acid. And we're going to talk about how that works in just a minute. And then lastly, it's a ribosome. Once everything is in order, the ribosome will actually fold the amino acids onto each other so it develops a shape, and then it becomes functional. The protein becomes functional. Because what establishes the functionality of a protein is its shape. Okay? All nucleic acids are constructed of some small molecules called nucleotides, and the nucleotides are composed of a 5-carbon sugar. We call them pentose, but they have names. The one in DNA is called deoxyribose, and the one in RNA is called ribose. Therefore, deoxyribonucleic acid, and RNA is ribonucleic acid. Okay? So it also has a phosphate group, and then it has a base containing 1 or 2 nitrogenous rings. Nitrogenous rings means that they have nitrogen in them. That's what it means. And they have names, and so here they are. Purines contain 2 rings that have nitrogen. So there's 1, there's 2, there's 1, there's 2. And the 2 purines are adenine and guanine. Okay? You're not going to be responsible for knowing how to draw these structures, but you will be responsible for being able to identify them. Okay? You won't have to draw them, but you'll have to identify. I'm going to show you how to do that. It's not hard once I show you. The other group is the pyrimidines, and the pyrimidines only have 1 ring that has nitrogen in it. So there's cytosine and thymine in DNA, and in RNA, it's cytosine and uracil. Do you see the difference between thymine and uracil? It's at that 6th carbon. You have, in uracil, you only have a hydrogen, but at that 6th carbon, you have a methyl group, and that's the only difference. You're not going to be responsible for drawing these things, okay? But you be responsible to tell me that uracil is an RNA and thymine is in DNA. There are rules for how these things pair up. So I always, in 9th grade when I was learning this stuff, I always remembered the preposition at, right? Adenine always binds the thymine. And if you know that, you can never ever make a mistake. Because if adenine binds the thymine, guanine must bind to cytosine, right? Has to be. That's the rule. That rule is never broken, okay? Therefore, we can do a lot with DNA, a lot. We can manipulate it. How often do you think we manipulate DNA, Bree?
All the time.
Okay, fine. Can you give me an example?
Like, you know those little Frenchie bulldogs?
Frenchie bulldog. Yeah, okay, good. Yeah.
And they look all fluffy
Yeah.
now?
No, we don't manipulate that by taking some DNA, put it in an organism. We just breed different animals. We can do that with plants too, right? So my wife, we have a big garden. My wife says, look at those cucumbers, they're huge. And look at these over here, they're little. So what she'll do is she'll go over there with the little Q-tip and she'll take the pollen out of the ones that are producing the big cucumbers, and she'll take them over and put them in the plants that are producing the little scrawny little cucumbers. And then we'll get seeds from that, from the fruit. And then next year, we have a whole bunch of big old gigantic cucumbers.
It sounds like your wife can get away with murder.
I can too. Not just but I don't want to. I'm not good at lying, so if I ever do the crime, I'm just like, just take me away. I get it. We're just not good at lying. We are big rule followers. And she's worse than I am. Like, she said, did you feed the cat? I'm like, yes, I gave him a quarter. She said, did you read my instructions? It said give half a can, not a quarter can. I'm like, oh, I'm in trouble now. Yeah, we just are big rule followers. The cat eats a lot, and he cries a lot, and so I've got to give him a little bit at a time to satisfy him, because if not, he'll just cry the whole time. She doesn't like that. She wants him to get whatever. Okay? So let me show you the backbone of DNA. So this is the backbone. I'm going to put a square around it, or a rectangle. That's the sugar and the phosphate. Yep, that's a backbone. So that's a backbone on that side. This is the backbone on the other side. Okay? So DNA always runs in an anti-parallel direction, and it runs from a 5' to 3' direction. Okay? let's talk about parallel and anti-parallel. Hejun, come here. Hejun and I, same here. We're driving down Mopac. You ready? Drive. This is, we're driving. Come on. And that's parallel, right? But if we're going in the opposite directions, I'm going north and south, I'm going north, you're going south, we drive, and we're going this way. That's anti-parallel, okay? So DNA, thank you. Let's give her a hand. DNA always runs in an anti-parallel format, and the direction for DNA is always 5' to 3'. So this is going this way, and this one's going that way. Yep. What does prime mean, Jeanette? What does prime mean? Oh, you know what prime means?
No, I feel like it's something to do with where it binds to.
That's exactly what it is. That's exactly what it
on
means.
yeah.
So 5' means that the functional group is attached to the 5th carbon. And this is not a carboxyl, it's a phosphate.
Oh,
It's that little P, that's a phosphate. So the rule is you start at the oxygen and you count clockwise. So here we go. 1, 2, 3, 4, and that point is 5. That's the 5' N, and that always has phosphate. Write it down. The 5' N always has a phosphate. Always. Okay? The 3' N always has a hydroxyl, an OH. So let's, down here, it's not showing it, but I'll show you one in a minute. But here's the oxygen 1, 2, 3. At that third location, there's a hydroxyl, an OH. Yep.
You said 3'?
3' N always has hydroxyl. That's correct. OH. Right. Good. What are these little dotted lines in between them? They're what? Not covalent. Hydrogen bonds. Hydrogen bonds. The thing about hydrogen bonds is you have 1 or 2 or 3, they're weak, but thousands or millions of them, very strong. But enzymes can break them down very easily. So we're going to tell the story of how the enzymes do that in just a minute. Okay? Good? So everybody knows that A binds to T and G binds to C in DNA, right? But in RNA, when DNA is being made, when RNA is being made from DNA, what binds to A? U. Correct. The rest of them are the same. G binds to C. Good? All right. So now you know more than 80% of the population of the United States, just from this one little hunk. Right? Not many people know this. Even today, if you ever, have anybody ever served on a jury? No? You have? You have? They don't like me at all. They're like, oh, Dr. Villarreal, how many degrees do you have? I said, 4. What have you done in your life? Well, I've worked clinical science, I've worked in the pharmaceutical world, I've done ecological work, and then they look at, we'd like to pass on him, and I get released very quickly. Never gotten past the first hour of jury selection. Always get released. They don't want somebody who can think. They want somebody who's going to be swayed by emotion. Because that's what they're going to try to do. They're going to try to sway you on emotion. My wife is even worse. She starts talking and she's out. She's gone. Really, go away. Okay? So, which one is weaker? Adenine or thymine or guanine and cytosine? Which one's weaker?
Adenine.
Yep. So there's more mutations on the adenine thymine portion of the DNA than there is in guanine cytosine. For instance, we talked about UV light last time. And what does UV light affect and what does it cause? It affects adenine and thymine and it produces a thymine dimer. Because adenine and thymine and guanine and cytosine. Okay? It's biological. Okay. So, there are two tenets of the central dogma theory, right? The central dogma theory, what's dogma? Lisette, what is dogma? It's our, no, don't just read it. It is our current understanding of how something works. Dogma. And dogma, scientific dogma, is forever changing. Once we get smarter, we change it, right? That's different than religious dogma, which has been codified in faith, and that never changes. Okay? So you're going, like if you're whatever religion you're going through all kinds of things right now. We're going through Lent and all kinds of stuff, right? And all that stuff has been codified, right? When does Ramadan start? Does anybody know? It's already started?
Yeah.
I wasn't sure, I thought it had, but it's closely related to the time of Lent. Right? Because we have similar beliefs. I don't get why people don't understand that we have more in common than we have that's different. Right? I just don't understand it. But especially when it comes to DNA, Bree, you and me are so similar, there's only, why do you look at me like that? There's only about 200,000 base pairs that make us different. And when you're talking about a DNA molecule, that's infinitesimally small. So there's only just a tiny bit that makes us different. Okay. So there are two tenets of the central dogma theory. The first tenet says DNA directs everything that goes on in the cell, including its own replication. Okay? And the second thing it says is that under the direction of DNA, RNA makes proteins. That's all it says. And so this is a really good image of what goes on in the central dogma theory. So
What's the
here's
second part?
under the direction of DNA, RNA makes proteins. It's written for you right here.
Oh, okay.
Okay.
Sorry.
It's okay. So here is the central dogma theory. There's a cell, the DNA gets replicated, and you have two cells, right? And both of them have a copy of the DNA. This is the other one where under the direction of DNA, proteins are made. Okay? There's this other thing over here called recombination. Recombination is really when DNA gets mixed up, right? And then you have a hybridization, which that's kind of a word that scientists use. You all would know it as a GMO, a genetically modified organism. How many people like GMOs? Am I the only one? So all of you all shop at Whole Paycheck, is that right? You don't go to HEB? So if you go to HEB, you like GMOs because 80% of all the stuff that they sell to eat at HEB is a GMO. 80%.
You tried those sweet Caroline blackberries?
The what now?
Sweet Caroline blackberries?
No, but I'm gonna do it now.
They're so sweet.
I know every time I go in there, I'm like, a pluot? What is that? Pluot? Let me try one. What? This is a grapple? What is it? A grape that tastes like an apple? Let me try it. Oh, by the way, it doesn't. Yeah, but it's a good marketing point, right? That
When
gets
they
you
say
to
it's
try things.
organic,
Huh?
they always say like organic cotton candy.
No, it's never gonna be
grapes, and I'm
organic
like,
cotton candy.
Ain't no way.
Yeah. No such thing as organic cotton candy. Can't be. Can't be. It's processed.
But
You
they're
can
allowed
have
to
a
throw
re-
that word around.
Well, I don't like the word myself because to me organic means it has carbon and hydrogen because I'm a scientist. Organic in that way is a marketing tool to get you to buy something that's a lot more expensive. Would you rather pay 50 cents for a pound of bananas or $1.20 for a pound of bananas? I'll take the 50 cents, thank you very much.
So does it actually matter like buying organic food? Like
Well, there was a study done that showed that the conventionally grown stuff and the organic stuff were equal in nutrients. There's no difference.
okay.
But the other thing that they tried to test was taste. But they couldn't find a good way to test that because everybody's taste is different. So they still say organic tastes better. And who am I to argue with that? It's your opinion about taste, right? Yeah. So I can't really argue that. So they still have that because, you know, you can't really argue taste. It's not, you can't prove that scientifically. It's taste. It's an opinion.
But I also feel like organic goes bad faster.
It will.
Yeah.
But do you know why? Because it's got a lot more organisms in it than the stuff that's been conventionally grown or radiated. Right? It does. Buy strawberries that were just normal strawberries. It'll last you a week, maybe 2 weeks. Organic strawberries, 3 days. Mushy, moldy.
And
You can still eat
it.
them. You know, blend them up in a blender and you won't know the difference. Nobody will know. Look at that.
Even if it has mold.
You, do you think, Sonia, that you don't eat mold on a daily basis? You do. You do. You do. You can't get away from them. They're everywhere. You can't get away from them. Okay. So, reverse transcriptase. Anybody ever heard of that?
Yes.
What's an, oh, Snazzy, you've heard of reverse transcriptase? What is it? RNA is used as a template instead of DNA. And that happens in only one thing that is on this planet, and that's the retroviruses. So this is HIV and feline leukemia virus. Yeah. So they use RNA as the pattern instead of using DNA as the pattern. Okay, so they use an enzyme that takes, instead of making, in the flow of information is usually DNA to RNA to protein. Here, you go to RNA to DNA and then make protein. So it's adding a step, but it's using RNA as a template to make DNA and then make proteins. You should know that it's going to be a multiple choice question on exam. Huh? What reverse transcriptase is. Okay? I'm giving you half the exam already. It seems like. Okay. Now we're going to talk about DNA replication of bacteria. So we're going to use bacteria as a model because they only have one chromosome. And therefore they're easy to talk about. Okay? Because the mechanism that happens in bacteria is the same thing in eukaryotes, except there's a lot more DNA in eukaryotes. And eukaryotic cells, when they undergo mitosis, there has to be at least 2 of them for anything eukaryotic. For bacteria, prokaryotic, they just need one chromosome. They're not going to go into mitosis. They undergo binary fission. Do you remember we talked about binary fission when we talked about nutrition and growth at the very beginning of the unit? You should probably understand how that works in general. Okay? So they contain a whole bunch of plasmids. So the plasmids are not part of the DNA molecule, they're extra, right? So they're going to go their own way to get copied, but they're going to get copied in a different way, right? And then it starts with enzymes, and the enzymes, the most important ones are called polymerases, and they're the ones that make all the copies. But we're going to talk about how these things work, right? So the DNA, whether it's in bacteria or in eukaryotes, is copied in a semi-conservative format. Right. So the DNA is usually balled up like this. It's very tight and wound up. It's grumpy. It doesn't want to unravel, so it needs something to help it unravel. And when it unravels, it relaxes. And then each of the strands get copied independently. And then each of those new DNA molecules, one containing an original strand and a new strand, the other containing an original strand and a new strand, they're going to go to make the new DNA molecules for the new cells that are being developed. We call that semi-conservative because one of the original strands of the DNA molecule is conserved in the new DNA molecules. Okay? So this is the way it works. The blue is representing the original strand. It's called the old strand here, but it's the original. And then the red is made. This is going to be for one cell, that's going to be for a second cell. So each of the DNA that's being made has got one original strand and one new strand, and both of those go to new cells. Right? We think that that happens, dogma, because that allows for less mistakes to be made when the DNA is being copied. being replicated. Okay? So the step of how this works is DNA replication. The DNA is unwound by enzymes. It's enzyme mediated. What does that mean, enzyme mediated? The enzymes are in control. Lots of enzymes, right? Complementary bases are added at the 3' end only. It cannot be added. The rule is it can only be added at the 3' end, right? The replication fork moves down. The newly replicated DNA rewinds, and the process is called semi-conservative replication. It's copied in a 5' to 3' direction, okay? And so let's talk about what that means, okay? So, the two strands have names depending on how they are moving, how they're being replicated, right? So you have two strands. One of them is called the leading strand, the other is called the lagging strand. The leading strand is oriented in a 5' to 3' direction, just like my little drawing is here. That's the 5' end, it's got a phosphate group. That's the 3' end, it has a hydroxyl. This is gonna be copied in the forward direction continuously, okay? Leading strand, always going to be copied continuously. The other strand is called a lagging strand, and the lagging strand is oriented backwards. So it has to have a different mechanism by which it is copied. It gets broken up into pieces, which we call Okazaki fragments, and then it gets copied, and then it gets put back together again, right? Because it's oriented in the opposite way, therefore it has to be copied backwards and then put back together again, okay? Lagging strand. The enzymes that are utilized for both the leading and lagging strand are topoisomerase, helicase, primase, and polymerase. But in the lagging strand, there are some others. Two of those are a nuclease or an endonuclease and ligase. Okay? With me? Okay. So, let's talk about how it works. So let's review. What does 5' mean? Huh? The phosphate is, remember that. Phosphate's always on the 5' end. But what does prime mean? The location, right? So again, if you take the oxygen counterclockwise, 1, 2, 3, 4, 5. At the 5' end is phosphate. The 3' end has hydroxyl. There's oxygen, 1, 2, 3. And then this is running anti-parallel, right? Good? But that's still the 5' end, right? Because if you count, you always count clockwise from the oxygen, 1, 2, 3, 4, 5. That's the 5' end. And here, 1, 2, 3, that's the 3' end. Even though they're inverted, you still count them in a clockwise format from the oxygen. Right? Good? And what do the little dots in the middle mean? Hydrogen bonds. So how many here? 3. How many there? 2. Okay? So, could you tell me what this is? Huh? This? What is that? I love your faces. Okay. Okay, listen. This is the way you do it. How many hydrogen bonds? How many hydrogen bonds? Ah, let's count them. 1, 2, 3. What base pairing does it have to be? Guanine and cytosine. Good. Now, is that a purine or a pyrimidine? Because they got 2 nitrogenous rings or 1? 2. It's a purine. What's the purine in the GC? Guanine. That's guanine. That is cytosine. Okay? Extra credit on the exam poo number 2 if you can tell me what these are. Good? All right. Let's go here. What base pairing is this? Amine. Why? 2 hydrogen bonds. What's that? Is that? What is that? It's thymine. Because that is the thymine, adenine, and then that's the pyrimidine, that's thymine. That is the purine, therefore adenine. Yep.
Are they always gonna be the same color?
No, they will not. As a matter of fact, when they get copied, they'll be grayscaled. Sorry. No knowing colors here. It's a sad day when you're out of coffee. I have to go to my backup tea. But no milk in this tea. Anybody got questions so far?
So you said guanine and adenine are purines, and cytosine.
Thymine and cytosine and uracil are pyrimides. Very good. Good? All right, here we go. So, let's talk about the way.
Ooh, I remember that picture.
You remember what picture?
The back, that one.
Really? Who showed it to you? Belzy Rosie?
Oh,
I like this one better. This is my favorite one.
there's a really good video online that he posted. I can't remember the person.
I got a couple that I like. I'll show them to you.
Okay.
Okay, I need a doc. You'll be my model. Okay. The doc is gonna come up here. I need an enzyme. Let me get one. Come up here, doc. You're a nice tall woman. Face your colleagues. And you're gonna, go back a little bit. You're going to be the DNA molecule. I want you to be all balled up and stressed out. Okay, good, perfect. Look at that. Look at that. Okay, so before DNA gets replicated, there's an enzyme. For those of you doing Levaquin, better pay attention. There's an enzyme that comes in, and it will touch and attach to the DNA molecule, and now the DNA molecule will relax. Don't open up, just relax. Okay, good, relaxed, all right? Once it's relaxed, because if it's super coiled, if you try to open it, it's gonna go back, right? It's gonna have a lot of torque in it. So now it's relaxed. Another enzyme comes in called helicase. And helicase, when it touches this, it opens up in a replication fork, way up. Perfect, look at that. Now you have the leading strand and the lagging strand. They're about to be copied, are you with me? So now, polymerase is like, I want to copy, but it doesn't know where to start. It needs help. So let's address the leading strand first. Another enzyme comes in and it puts a little tiny piece of RNA that's called a primer. It's the enzyme that does that is called primase. And when it goes there, polymerase goes, oh, that's where I go. And it goes, makes it copy continuously like that. About 1,000 nucleotides per second. Amazing. Okay? Now, the other side is more difficult because it's oriented in the opposite way. So now there's an enzyme that comes in and it nicks it. Tick, tick, tick. Makes some different pieces. Those are called Okazaki fragments.
Oh, no.
What do you mean? You want to call them something else?
I'm gonna remember all this.
No, you're not going to remember. You're going to know.
Okazaki.
There's a
no,
different
no.
Okazaki over the famous scientist who came up with the concept.
Okie dokie.
No, Okazaki. Not okie dokie, that's wrong, Okazaki. So now it's got pieces, and primase comes in and it puts a primer in each of the pieces. Yeah? Polymerase goes, okay, my turn. Copy, 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 copy. And then the last enzyme that comes in is called ligase. And ligase, when you ligate something in science, you put it back together. You glue it back together. So it comes in and it goes, oh. And it puts it all back together. Now you have 2 copies of the DNA molecule. Yeah? Let's give her a hand for really. So this is my favorite image. I'm telling you, this image in some fashion is going to be on the exam poo. Bree, I'm gonna send it to you.
I know, but for
And
me to remember.
it's already in the PowerPoint, but I'm gonna send it to you. Okay? So let's just practice. What is number one? That's something that relaxes the DNA molecule. Topoisomerase. What is number two that relaxes the DNA molecule? Oh, I'm sorry. Oh, my bad. Number two is what opens up the DNA molecule. Helicase. Mm-hmm. Don't worry about three. Those are small binding proteins. I didn't talk about them. It won't be on the exam poo. Okay? Number four is the light pink thing. And number four is putting these pink things in the actual fragments. What are those pink, those dark pink things, those fuchsia pink things called? Primer. But the enzyme that's putting it in, the light pink thing, is called what? Primase. Oh, you remember that from 1301?
I've been studying, that's
Oh,
why it's
yeah.
late.
So then polymerase goes crazy. It copies here. Is this the leading or lagging strand? How do you know that?
It's continuous.
Continuous. But how about these guys? Is this leading or lagging strand?
It's lagging.
Why?
It's broken up.
And what are those little fragments called? Okazaki fragment, not okie dokie, Okazaki. Okay? We pay honor to Dr. Okazaki for doing that. And then at the very end, we have something that comes back and puts it all back together again. What is that called? How about if I send you this?
Oh,
With all the answers on it.
yay.
But it won't be, the answers will not be on the exam pool.
I know you have to remember it. I mean, you have to know it.
That's better than memorizing. You have to know it. I'll send it to you. I don't know what y'all are taking pictures of.
It's all a PowerPoint.
Yes, I know. But listen, everybody's like taking pictures.
I'm
What's
writing
going
down the
on?
explanation.
Hey, why is this hard?
I feel like I used to know it.
How about you will know it?
Yeah.
Used to is past tense. I want now, present. Let's review. What does semi-conservative replication mean?
One of the strands, the original strands, will be conserved in.
Nice, Justine. Very nice. Old Justine is ready.
I haven't done a rabbit hole this the first time I learned it, so.
How do we know it's the 5' end? Huh? Don't forget that. Know it forever. If I catch you at HEB, I'm gonna quiz you. And by the way, I'm gonna be in Bastrop this weekend. You better watch it. I'm coming in.
Do you go to HEB every day?
Almost every day. Almost every day. Because we don't buy a lot of stuff. We buy a little bit at a time.
Yeah, I'm the same way,
Okay.
yeah.
Like tonight, I'm making chicken.
So you're a recipe shopper.
No, I'm a hungry chopper. I walk around whatever I want. That's what I'm gonna get. So yeah, but how do we know it's the 3' end? Hydroxyl. Remember to know that it's on the exam. Okay. What does topoisomerase do?
It relaxes.
Do you know how Pejun? Do you know how Levaquin works? What is this? You look like a little duck. Levaquin blocks topoisomerase, so the DNA never gets relaxed, so it never gets copied. Isn't that cool?
What do you mean by relax?
So we say that the DNA molecule is super coiled when it's wrapped around, it's super coiled, and when if you opens up but nothing relaxed, it goes back right away. So topoisomerase relaxes so it can open up. Okay? Good? Questions? So the things we're covering today will help you if you're doing anything that interferes with the DNA molecule. That's going to be the fluoroquinolone Levaquin, That's gonna be metronidazole. Works a different way, but it works on the DNA molecule. And let's see what else. I think that might be it. The other two are proteins. Oh, Bactrim. Bactrim also, who's doing Bactrim? So Bactrim also works on 2 steps of the nucleic acid making. Okay? Good? Alright. So I'm gonna show you a video, and I want you to reflect on how cool this stuff works. You know, I can explain it with words, but if you watch a video, a lot of times it makes a whole lot more sense. This is one of my favorite videos. There we go.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
Pretty cool.
The sound of disruption.
Like, I have a question.
Yep.
Like, how do they see that? In a microscope?
No. We would never be able to really fully see that. So a lot of this stuff is
Computer generated.
done from our understanding of how the enzymes work. And so we make something that makes sense for us. Do we have it perfectly aligned with the way it works? We don't know. But this makes a lot of sense to us from a scientific perspective. So therefore we follow it. It's what's called dogma. That's why you have your central dogma theory. This is our current understanding of how it works. Okay? Will it change? Most certainly. Because we're getting smarter every time. Every year we get smarter. And something new is added. It's really hard to see something that is at the nanometer or size, right? Because a microbe, a bacterium, is a micrometer in size. These things are smaller than that. They're nanometers or picometers in size. There's no way you can see those. Maybe with the electron microscope that UT has, but you could never see this happening in real time. We can show you how mitosis works because we can stain the DNA as it's being separated out. But mitosis is really the end of the DNA being separated out. The copying, the replication of DNA has already happened in the S phase of mitosis, or S phase of cell cycle.
Want to strengthen
Mitosis.
your math and computer science.
Sorry, that was me.
Was that somebody?
me,
who was,
sorry,
okay,
sorry.
I was like, did I leave that thing on? Okay. Oh, so it's our current understanding based on a lot of people's research. It's beautiful. Really,
It's
just.
interesting to me, like, how they come up with this.
Oh, we know a lot of other stuff too. Huh? I know, I can't spend all day telling you about everything.
Okay.
I can only tell you a little bit of stuff. Okay, let's go on to talk about RNA, the way RNA works. So, the 3 types of RNA are messenger RNA, transfer RNA, and ribosomal RNA. And they have functions. What does messenger RNA do? Makes a copy
Makes a
of
copy of the DNA.
DNA in RNA language. In other words, there's no thymine, it gets to be yours.
Yes.
Okay? What does transfer RNA do? Translation, so tell me what that means. I agree, transfer, tell me what that means.
Because there's a thymine that it has to translate into another.
An amino acid.
Yeah.
So it takes a look at the information that is in a messenger RNA, and those DNA and RNA are just codes. They're just codes. And they code for an amino acid. Transfer RNA looks at that, says, oh, this is what it is, and it brings the amino acid, right? Which now brings up the term codon. Codon is 3 nucleotides, and the 3 nucleotides code for 1 amino acid. Okay? Are you with me? A codon is 3 nucleotides, and the 3 nucleotides code for 1 amino acid. And it's going to make sense in a minute, I promise. Okay? And what does ribosomal RNA do? It processes whatever the amino acids are in that chain, folds it up, makes it so it has shape, and now the protein has function. Because what establishes the functionality of a protein is its shape. Isn't that cool? Amazingly cool. Even cooler than the replication process. Now, some of you all are going to confuse DNA replication with protein synthesis on the exam. Don't do it. Think about the fact that when we're talking about DNA replication, we're talking about DNA being made into new DNA. Protein synthesis, you're taking a strand of RNA and making proteins. Okay? You've got to be sure that you separate those out. Every semester there's always 1 or 2 people who get them really confused, and then the exam is wrong because of the way you're talking about it. Okay? Okay. So, can you appreciate that this is your transcript? So messenger RNA, when it gets made, it's also known as a transcript. Those are, that mean the same thing. But then it gets copied, so DNA is made into RNA. Can you appreciate what binds to T? A. What binds to A?
U.
U. What binds to C?
G.
So, and then you can see how it's made, right? There are no, if this is DNA, this is RNA, there are no T's. There are no thymines in RNA, only in DNA. Okay? So, this is a eukaryotic cell. Look at the complexity of what's happening. Here's the nucleus, there's a pore, here's a DNA molecule. One of them is, RNA is copying it into RNA language. There's a bunch of things more that are going on that I'm not going to talk about, right? That's for a different course. But the messenger RNA leaves the nucleus through a pore, and once it gets there, it complexes with the ribosome, right? And then the code in it is called a codon. 3 nucleotides equal 1 codon, which codes for 1 amino acid. These little firecracker looking things are transfer RNA. They look to see what the information is in the messenger RNA, and then they have what we call an anticodon. An anticodon is complementary to a codon. Therefore, there can never be a mistake made in which amino acid gets placed in the series in the growing polypeptide chain, because the anticodon is complementary to the codon. Okay?
Anticodon, they can never be a mistake.
An anticodon is complementary to the codon, therefore there can never be a mistake in the amino acid that's placed in the growing chain of amino acids. Okay?
Mm-hmm.
Yep.
What causes a misfolded protein?
Oh, a lot of things.
Well, I know other than it can be like environmental factors and stuff like that, but
So what, like,
in that process, is there?
okay, so the biggest one that can cause misfolded proteins is smoking or vaping.
Does that have a mutation?
Huh?
Does it have a mutation?
Partly, yeah. The mutation causes misfolding. Yeah. So there's a bunch of things that can happen. I mean, too much sunlight. I mean, there's just a ton of things, right? But the biggest factor that affects us as humans is smoking and vaping. And people always tell me, well, vaping is much better for you because there's no smoke. I said, no, not right. I said the vape is actually an oil, so the oil takes the carcinogens right into the cells, so vaping is worse for you.
That's what creates popcorn ones.
It creates what?
Popcorn ones.
I don't like that term, but yeah, okay. It makes it funny, and I don't like that. I want it to be serious when you're talking about cancer. Yeah. Okay?
No, it's just only because they say it looks like popcorn. I guess that's why. Wow.
Okay, let's talk about this, right? So, protein synthesis happens in 3 stages. It happens in initiation, elongation, and in termination. Okay, so let's talk about this. Initiation always starts, let's just go with this. What is this called here? Okay, what is this thing called? Huh? mRNA, good. What is these 3 things called right here? And what are those 3 things called right there? What is this thing here? And what's that thing? Good. Why is this hard? It's that easy. Don't make it hard. Okay? So, the start sequence or the start codon of protein synthesis, and it'll always be like this with me, is always going to be AUG. Adenine uracil 1. Always. So when I ask you to do this on an exam pool, and I will, if you don't have AUG as your first amino, as your first codon, you better go back and look. Something happened. Okay? So once that happens, then protein synthesis is about to be started. That's the start codon. Always. Okay? Then in elongation, you have all these other codons coming through, coding for an amino acid, and all of these amino acids are being put in a chain. At some point, there's a termination sequence that happens, comes in to the actual ribosome, and that those termination sequences, there are 3 of them. They're UAA, UGA, and UAG, and they're one of those 3 all the time. You should just know those by heart. Just know. Okay? When one of those comes in, they don't code for an amino acid, they code for done. We're done. Stop. Okay? So, what's a codon again? What? But before that, 3 nucleotides which code for 1 amino acid. So here is my favorite codon table. And the way you engage this is you always start on the left. The left is your first base. Then you come up top, that's your second base. And then you go over here up to the third base. So let's just do AUG, right? Here's A, here's U, bring it down and bring G over. AUG codes for methionine, which is a start codon. You can write in methionine, or you can just use the 3-letter abbreviation, which is MET. Okay? Either one of those is fair. Let's practice using the codon table. What does GGU code for? What do you think? GGU? Glycine. Good. What about ACC? I put this here to be funny, but ACC is Austin Community College. But what is it?
309.
ACC is 3 and E. Are you ready? Here we go.
Wait,
Boom.
wait, how did you find, oh, yeah.
ACC. Look. Here we go. A, C, C. 3 and E.
Oh, I did it wrong.
Don't do it wrong.
I know.
Get it right. What about TAG? What about TAG? What about TAG? Huh? No, TAG. What about TAG? Tell me what you know.
True question.
It ain't a trick question. I'm testing your total knowledge.
Or it would be A.
It cannot be anything because there are no thymine in RNA.
So what is the trick question?
I don't do trick questions. I'm testing your total knowledge. Alright, so if I give you a strand of DNA, how do I know it's DNA? It's got T's. Could you give me the transcript? This is yes, this is no, this is not allowed. Only this. Okay? What do you mean? Why are you looking at me like that?
Hold on, I'm zooming in.
Okay, here we go. Oh, you and Christy both need glasses.
And me.
Oh, and Bree, like,
I think because my brain hurts, okay?
Yeah,
That's all it
okay,
is.
here we go. So give me the transcript. Are you with me? Go. AUG, and if it's AUG, you know what? It's a start codon, and you're good with me. Because in another class, not here, in another class, I would do things whacked out. I would mess them all up. They had to find out what the start codon is. For you all, it's always going to be start codon. Always going to be AUG. Okay, here we go. Ready? Let's go. Why is this hard? Boom. There you go. Good. Now, could you give me the amino acids? Okay, that's good. AUG stands for what? Methionine. What about CUA? Huh? Leucine. Okay. What about GGA? Glycine. Good. What about GGA? What about AUU?
Isoleucine.
Isoleucine. Very good. And then UGA?
That's gonna be serine or arginine.
No.
No.
UGA.
Stop codon.
It's a stop codon.
Oh.
Okay, now, boom, there you go. How many codons are in this fragment? 5. How many amino acids does it code for?
4.
Oh.
Stop.
The stop codon doesn't code for an amino acid. Do not fall into the trap, or what Jeanette likes to call the trick question. It is not a trick question. I'm testing your total knowledge. Okay? Yeah, you're gonna have to be able to use this. I will give you a codon table on the exam you put. Okay? Sonia, why are you? I'm gonna show you one last video and then we're gonna call it a day. This one's a little bit more monotone.
Prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formylmethionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has 2 sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a 3rd site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formylmethionine binds to the P site. A transfer RNA that recognizes the next codon and carries the 2nd amino acid then moves into the A site. The formylmethionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of 1 codon, and the transfer RNA that carried the formylmethionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of 1 codon. As the shift occurs, the 2 amino acids on the transfer RNA in the P site are transferred to the new amino acid and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
You wanna turn the lights back on? Okay, I have a few more things to talk about, but we will talk about them on Thursday, which is gonna be mutations. It will take me 10 minutes to get through it, and then that'll be the end of the material. So, we will then begin to talk about fungi, but the last 20 minutes of our class will be for review for the exam next Tuesday, okay? Anybody have any guess?
Do you have Haley's having any as
She is, but the morning class has decided to take the exam after spring break. They're crazy. I mean, y'all can do that too if you want. They think that they're gonna spend time studying. They're not. Yeah, it comes up pretty quickly. So, but, you know, I try to make as many people happy as possible, and one of them said to their colleague, I guess they were like doing it behind my back and sneaky, and so there was enough of them that they voted to take it after spring break. So I'm gonna adhere to that. There's a couple of people that are going on a vacation. Well, I can help you. If you want, I'll just give you the exam, or you can come take it with you all. And so I'm gonna make an adjustment for them, because I don't want people to miss out on their vacation and have to study during and some of them already have another test coming back from spring break. You all have the same option if you want. If you decide Doc, don't be shaking your head till I finish talking. So, but y'all can have the same, you all are the same. So if you all decide, like the majority of you decide you wanna take the exam after spring break, I will do that. But it has to be a vote in that case. So y'all can talk to each other if you wanna do that. I gotta be honest with you, I don't recommend it. I told them that, but it was their vote, and 11 of them voted for yes, and so I'm gonna abide by that. It's up to you. Y'all have some negotiation power when you take the exam. I'm gonna tell you now, very few of you will study over spring break.
I don't want
I mean, no, I'm just saying. My 1406 guys did the same thing. They want to do it after spring break. I never had that happen.
That's stressful.
Well, I mean, yeah, but what's gonna happen is most of them won't think about this until when? The night before. Oh, I have an exam! And then they're just gonna be whacked out. Okay? Anybody else have any questions? Okay, how about if y'all take 10 and we'll see you in